Thank you. Yeah, I'll segue into my talk. Briefly, I'll tell you that at noon, we're going to have a break. We have two non-CME speakers at that time, so you can kind of relax and watch those. But I think we should just keep going through the talks. As you know, my name is Dr. Charles Cobbs. I am a neurosurgeon here. I have been involved with the Ivy Center here since I came 11 years ago. And I'm very fortunate to work with a great group in the lab and a full team of neuro-oncology and Parvi Hoti, who gave a talk earlier, and our foundation folks are here, and I really appreciate everything they do for us. I guess I'll start here. Let me go back. Okay, let me go back. So this is a picture I took out of my window the other day, which is reminds me that when you can actually see clear skies, more or less, it's an amazing view. It's hard to believe that was 7 a.m. like 2 weeks ago. So you've all heard a lot. I'm very biased toward immunotherapy as being the way we may be able to cure glioblastoma. I think what I'd like to do is go over a few of the concepts that you've heard about, because we've been flying through things, and I think for this particular audience who may not be so sophisticated in cancer biology, it'd be good to just sort of reemphasize general concepts. So I'm going to go over some concepts and then try to pull it all together as to what we're trying to do in our lab. So we know that checkpoint inhibitors are things you hear on TV ads for, which are curing certain cancers like melanoma and lung cancer. But why is this not working in glioblastoma? The concept essentially is that in where's this guy? How do I do this guy? Oh, is that right there? Okay, yeah. So in a normal cell, your body knows what is a self antigen and what's not self. And so your T cells will circulate, they'll see your own proteins, and they'll not kill that cell. If a tumor cell has a lot of DNA mutations, that'll cause an RNA mutation, which will eventually encode for a new amino acid, which will eventually encode for a new shaped protein. And your immune cells can identify that as different. Unfortunately, when cancers evolve, they develop these, the T cells have these inhibitory molecules, so the cancer can grow and then kind of become exhausted and not kill the tumor cell. However, the discovery of these checkpoint inhibitor drugs that you see the names here that block these pathways revolutionized cancer immunotherapy by putting the brakes on the T cells, unleashing the T cells to now identify these foreign antigens due to mutations. So tumors that had high levels of mutations became susceptible all of a sudden to this awakening of your T cells to kill them. But if you look at this scale of logarithmic number of mutations in different tumor types published about 10 years ago, you see that certain tumors like melanoma have 1,000-fold more mutations than low-grade gliomas, for instance. And it's only these very, very highly mutated cells that have so many altered RNA mutations that lead to new proteins that allow your T cells to actually do the surveillance and kill them. Unfortunately, glioblastoma is considered a cold tumor, and the tumor cells shown here in this blue cell produce a range of things that block the function of immune cells. And you'll see in a later slide of mine that cytotoxic T cells hardly get into the tumors at all. And without the T cells coming in to kill the tumor cells, immunotherapy is not going to function very well. And a lot of that has to do, as you've heard earlier, by certain macrophages and mononuclear cells that produce all these cytokines that are inhibitory to the immune system and block the ones like IL-1 and tumor necrosis factor that can activate an immune response. I'm going to focus this talk on novel antigens and two sort of thematic pathways that we're interested in: PAMPs, DAMPs, and TAAs. So PAMPs are pathogen-associated molecular patterns. DAMPs are damage-associated molecular patterns. And these are things that you can try to activate or enhance in the tumor to get your T cells to come in and see the tumor, even if it hasn't really paid attention to the tumor previous to that. And tumor-associated antigens are antigens or proteins that may be associated with the tumor and not in normal cells. And if you activate these pathogen and damage-associated molecules, you can enhance T cell activation. T cells may go back to the lymph nodes, train other T cells to come back to the tumor and then kill the tumor cell. So what antigens do we know you can attack in glioblastoma? Well, we know certain antigens are mutated, like EGFR, V3, and IDH1, but efforts to eradicate glioblastoma by just targeting these single molecules have been unsuccessful because the tumor will often just mutate and grow out of that. Certain molecules are overexpressed in we've tried to attack these with CAR T cells also, and the same thing has happened. My lab has been interested in viral antigens associated with glioblastoma, and we discovered cytomegalovirus antigens were highly associated with glioblastoma. So we're continuing to look into that, but we've also started looking into another phenomenon called alternative splicing, which can lead to tumor-associated antigens. And then we've recently begun a collaboration with a biotech company that has immune activators to activate those damage and pathogen-associated pathways in tumors through sort of a gene therapy or vaccine type approach, if you will. So my group discovered cytomegalovirus was in tumors, and this has been taken up by folks at Duke and other institutions, and you'll also hear later today by Dr. Rajiv Khanna in Australia, how targeting CMV may allow us to target glioblastoma. And there's been interesting success in some preliminary trials by immune therapy to cytomegalovirus, either with activating T cells in patients by activating dendritic cells and giving them back T cells. And here you're seeing these red cells are cytomegalovirus hypothetically infected tumor cells, and if the immune system comes in and kills those, it may reduce the tumor burden, or it may actually cause even a more heightened immune response with other immune cells to come in and take the tumor out. And also, if this process occurs in the setting of tumor cell death, you get the damage-associated features, and you may get off-target killing of tumor cells. So by activating an immune response to a pathogen in the setting of tumor death, you may release tumor molecules, and there may be some epitope spreading and cross-reactivity to kill the tumor more actively. So hold that thought for a second. About 5 years ago, a postdoc in the lab, in our lab, read a paper and got interested in this concept of alternative mRNA splicing. Here you see Dr. David Baltimore, who Dr. Ashish just mentioned a minute ago, got the Nobel Prize for retroviral discovery. So what is alternative splicing, and what does that have to do with tumor antigens? Well, we know that DNA encodes for RNA, and RNA is made like a string of beads, is like a string of beads, and only certain portions of the RNA called exons are strung together, and these pieces of RNA will then encode for protein. So in your normal adult life, you may have a protein shown here that is encoded by this DNA, which is only incorporating A, B, and D portions of the RNA, or exons. We know, however, that during development and sometimes in cancer, there's alternative splicing, and so instead of doing the normal A, B, and D in cancer, this RNA may be spliced together, including C. And so if this portion of the RNA molecule is incorporated only in the tumor, then it would encode for a protein that has a segment that your immune system has never seen and is not used to seeing. So our lab started to investigate this, and we looked through RNA from different glioblastomas, and we found that there were hundreds and hundreds of potential epitopes based on alternative splicing, or areas that could be targeted if we could find proteins encoded by that, because the goal is to find immune targets that are specific to the tumor but not seen elsewhere in the body. And this required us to use a novel RNA sequencing technology using equipment from Oxford Nanopore, which does long-read sequencing and doesn't do the normal sequencing, which has little small pieces, then you can have gaps. These very long reads will give us huge areas of RNA that are sequenced, and then we can say which portion of this is not normally seen in normal. And the reason I say that is because I have a routine practice when I take out a glioblastoma, especially if it's a deep tumor and I have to go through the surface of normal tissue. I take a small piece of normal tissue and we'll sequence the RNA from that and the RNA from the tumor from the same patient. And at the same time, we'll also take protein from the normal area and the tumor. And this is a polyacrylamide gel. And the reason I'm showing you this is if we have a tumor and the normal from the same patient, if we run the proteins of all these, of the tumor and normal and sort them by size, what we've just done is we've taken little pieces from different sections and we started working with an investigator who's now at Ohio State Immunotherapy Cancer Research Program. And he does mass spectrometry, which breaks these proteins up into little pieces and then analyzes all of them. And so we can compare on a computer side by side. If we find genes in the RNA that we think are going to predict for a novel protein, we can directly look for those. And in our lab, we had a brilliant pre-med student who's now a student at Yale who wrote a software program that can compare the normal and tumor by going through all these amino acid areas that have been broken up and read by mass spectrometry. And for instance, in this case, we had 3 normals shown in blue, and next to them were the 3 tumors from the same patients, 1, 2, and 3, side by side. And this software has allowed us to compare, go through the whole protein analysis of normal and tumor and compare. And we found there are certain proteins that are highly expressed in the tumor that we don't see in normal, that are normal proteins that we knew about. And for instance, HMOX1, which we published previously, is highly expressed in glioblastoma. But then there are other ones that we've discovered based on our RNA analysis and then looking for them in the protein, such as this gene MSI2. And you can see 3 normals, 3 tumors, 3 normals, 3 tumors. And these chunks of protein here are from alternative spliced areas of the gene, and there's no expression in the normal, and it's high expression in the tumor. And we showed this even further when we looked at normal from the patients and then a bunch of tumors where we see this protein fragment is expressed in most of the tumors but not in normal. And that leads us to the potential of targeting these novel tumor-associated antigens from alternative splicing. So to summarize this concept, which was in this original paper by Dr. Baltimore and colleagues, if you take tumor and normal tissue and you sequence the RNA, you can ask the computer to identify areas of RNA that are expressed in the tumor, but you don't see them in the normal. Then you tell the computer to look for those, the proteins for those sections of RNA in the mass spectrometry data, and then you can come up with hypothetical novel antigens for your immune system to attack. So we're right now in the middle of doing this with a colleague at Ohio State, Dr. Brian Searle, and we're doing huge amounts of sequencing and mass spec to look for novel epitopes. Hopefully we'll find some that are on the cell surface that we can make antibodies to or T-cell receptor specific molecules to, and then we'll have to test them downstream, and I'll get to that in a minute. I'm going to transition slightly to show, you heard from Dr. Chioka earlier and talking about oncolytic herpes virus. I was fortunate to be involved in one of the manuscripts we put together where we took glioblastoma and made slices of this to see if, when he put the viral infection into the tumor, if the patient's immune cells could come in to attack the tumor cells. So my lab has taken that concept and we've got that up and going now. So we're going to use this model system to further investigate whether we can make these cold tumors hot, and I'll show you how we're going to do that. Basically, when I take a tumor out now, we put it in a thing called a compressome, and then we slice these thick sections that are 350 microns thick, about 35 cells thick, and we put them in culture, and they grow, they live in a artificial spinal fluid environment, and they can live for several days. And if you stain them, this is a glioblastoma we did recently with H&E. You can see the tumor here. And we've started analyzing them for evidence of infiltrating cytotoxic T lymphocytes. And here you can see very few T cells are in this glioblastoma. There's some around the blood vessel here. But what we're doing now is we're establishing a model system so that once we identify potentially new tumor-associated antigens from the mass spectrometry, we can make antibodies to those and see if we can target them in the tumor, or see if we can vaccinate the T cells from that same patient in a dish and then have them attack the tumor cells in this model system. And one other thing that has been pretty exciting to me is we have now a collaboration with a biotech company that's near the Fred Hutch here called HDT Bio. This biotech company has traditionally been in the space of RNA vaccines for infectious diseases. And Peter Berglund and I met a couple of years ago, and I started talking to him about, they actually had a COVID vaccine, kind of like the Moderna vaccine that was used in India. And he's got the patent for this lipid complex that is encoded, encased with RNA that can be delivered specifically to cells, and then the RNA will start pumping out the gene of interest. And I said, what if we tried to vaccinate the tumor itself? And this kind of goes back to what Dr. Chioka was showing when he was vaccinating the tumors with the herpes virus. What if we could vaccinate the tumor with viral antigens and things that activate this innate immune system? And he said, well, we have a molecule that is similar to the effect of something that's used in the cancer community all the time called poly-IC. Poly-IC is a synthetic RNA, double-stranded RNA, and if you put that in a tumor cell, it will make the cell highly activate something called toll-like receptor 3, which is a pathway that tells the cell that there's a very angry viral infection going on. So it tells the tumor cell and the whole environment that there's a viral infection, and it causes the tumor cell to pump out cytokines that activate cytotoxic T cells, and it also causes the tumor to die. So putting all this together, there's this new concept emerging that's only a few years old that Dr. Chioka's work, I think, alludes to significantly, that when you look at these cold tumors, is there any strategy that we can do to overcome the immunosuppression? And there's a group that published a paper a couple of years ago where they put viral peptides in the tumor microenvironment, and they elicited an immune response, just like Dr. Chioka showed that patients who'd already been exposed to herpes simplex 1, if you put that virus in, that recruited an immune response that helped kill the tumor. Well, this group has been putting peptides of certain viruses like cytomegalovirus in the tumor and reactivating this immune response. So we were awarded a small grant from the Paul Allen Research Group here to basically set up this system so that we can put the glioblastoma slices in culture and add in the patient's own immune cells. And then we will either give the gene product for toll-like receptor 3 plus or minus cytomegalovirus peptides to see if we can cause the tumor slices to become infiltrated with cytotoxic T cells. And then we'll use a very elegant system from a company here in Seattle called NanoString that will allow us to analyze areas where the T cells have infiltrated to see if we're actually getting a robust inflammation and immune response. Here is an example of a preliminary experiment we did with this company. That this is this lipid emulsion RNA molecule that produces something called GFP, which is just a fluorescent molecule. So this is a case we did just recently of a glioma where we took the tumor out and took it to the lab, injected it with a needle with this molecule. It spread out throughout the tissue. We let it sit overnight. Then we sliced it into these thick cultures, thick slices, and we showed that on fluorescence, it's a little hard to see, but there was significant expression of our molecule of interest, in this case just a marker. So the hope is that if we could get to a point where we could inject the tumor with something that activates an immune response plus some viral antigens, we may be able to get a very robust infiltration of the tumor with cytotoxic T cells. And hopefully in the same process, we'll be able to use our discovery of the tumor-associated antigens that seems to be moving forward from the alternative splicing project I mentioned. So a lot of stuff going on, but in summary, we have an ex vivo, which means right out of a patient's system for studying immunotherapy treatments in glioblastoma patients using their tumor and their immune cells, and we're going to modify their tumor in the dish and see if we can activate their own immune system to attack it. By doing that, we're also going to hopefully identify some novel tumor-associated antigens that we can target these slices with based on the alternative splicing. We know we can amplify an anti-tumor response by calling in memory cytotoxic T cells, and we think if most people have been exposed to cytomegalovirus, for instance, if we can vaccinate the tumor itself and get a T cell response to the CMV infection, that would be exciting and sort of analogous to Dr. Chioka and herpes simplex 1. And finally, if we do that in the setting of spreading a signal that there's a viral infection with this molecule from HDT Bio that I mentioned, it may really amplify the signal to increase an immune response. So the goal being anything we can do to get those T cells to infiltrate the glioblastoma, and then hopefully there will be epitope spreading like was shown by Dr. Chioka's lab, where not only will you kill the infected cells, but the other cells, all of a sudden the immune system will target as well. So hopefully this will lead to some exciting preclinical work that will lead to clinical trials in patients. So someday I hope my vision would be that we would biopsy a tumor and then inject at the same time some of these molecules to incite a massive immune response to the tumor and have an immunotherapy treatment. Thank you so much. I'd like to thank Mohsen Karimi, who did a lot of the alternative splicing work, and Parvi, who you heard from before, the folks in the lab, Jae-Jun Yoon and his wife Lee, George Sun, the medical student, Brian Searle, and this was funded by R21 grant from the NCI and the Paul Allen Research Center Award. Thank you.